we do not like any other pollen to settle on the stigma and finish the process of pollination and then fertilization and so we want to produce these superior varieties or hybrids, right? It can happen only when we know both the parents. We should know the female parent and also should know the male parent, and pollen will be selected from plants that have some superior qualities, including the female too. So the female and the male, whatever the qualities, the good qualities they have, those good qualities will be combined and form a new variety, and that is what we call it a hybrid. Right. So first, in the process of artificial hybridization, we are going to remove the anthers with the help of forceps. Once that is finished, we call that flower as a emasculated flower, and that emasculated flower will be covered by a bag, mostly like a butter paper, right, to prevent contamination with the unwanted pollen. Now once the stigma comes to when stigma comes to receptivity, then pollen from desired plant is dusted onto the stigma, thereby completing the process of pollination. So once the pollination is completed, the flower is again rebagged. The flower is again rebagged till it forms the fruits or seeds. So this is how we perform artificial hybridization. So now this is all about the pollen pistil interaction. Right. The next one we shall see about double fertilization. We'll see about double fertilization. So definition: a process that involves syngamy and triple fusion in the embryo sac of angiosperms is what we call a double fertilization. So this is a process that involves two events. As two events are involved, the process we call it double fertilization. So here syngamy is fusion of male gamete with the egg. So this we call it syngamy, and triple fusion is again the fusion of male gamete with the secondary nucleus of central cell is called triple fusion. As these are the two events involved in this process, we call it double fertilization. So now let us see how the double fertilization actually takes place. So here the stigma, okay, and the ovule here, right. And inside that the embryo sac. So you may find here, okay, this is a chalazal side, and we know here these are, okay, antipodals, and this side is the micropylar end, okay, and here you will find, okay, the synergids and the egg. So here is the egg cell, okay, and these are synergid cells, okay, right, and you may find here the two polar nuclei. So once the right type of pollen settles on the stigma, okay, so we know that there is, okay, outer part of the pollen grain is the xray and the inner part is, okay, intine, and that has, okay, a large vegetative cell in which embedded is a generative cell. And we have already studied that in majority of angiosperms the pollen grains are released, okay, at the two-celled stage. So at that two-cell stage the pollen grain will have one vegetative cell, that is a large cell, and in that cytoplasm of the vegetative cell will be the generative cell, which will be smaller, okay. So here a compatible pollen, okay, settle on the stigma. So now this undergoes the germination, and the germination is a process, okay, where the pollen tube emerges out through the germ pore, okay. So here the pollen tube emerges out from, okay, germ pore, and we call this process as, okay, pollen germination, okay. Now you can see here, right, the pollen tube emerged out. So this is the pollen tube, and you may see so many pollen grains that might have settled, okay, on this stigma, and most of them may be, okay, a right type. So here I am showing it, and in some, okay, so you can see the pollen tube, okay, and in this case just it emerged out. So you can see the pollen tube is of various, okay, lengths, right. Only the one which grows first, okay, may come and fertilize, okay, the embryo sac, okay, or the ovule, right. So here inside this the generative cell, okay, if it is released in two-cell stage, the generative cell, okay, undergoes mitosis and produces two male gametes. So I call them, okay, sperms. So two sperms will be produced here. So generative cell, okay, undergoes mitosis and produces two male gametes, and you may have here, okay, the two male gametes, and this is, okay, the pollen tube, and you may find even, okay, tube nucleus. So this is the tube nucleus, and you may see here these two are, okay, the male gametes, okay. You may find in almost, okay, in all those pollen tubes there may be so many pollen tubes, okay, developing, and then they all pass through this, okay, style. We know this portion of the pistil is the stigma, and this portion is the style, and this is the portion ovary, inside which we have is a, okay, ovule, and inside the ovule is the embryo sac. Okay, so now the pollen tube grows through the style, and then, okay, finally, right, so I'm just showing with a dotted mark, so it comes in this way, okay, right. So here, and then it finally, okay, enters through the micropyle here. Okay, so if the pollen tube enters through the micropyle, okay, if pollen tube enters through the micropyle, the condition is called porogamy. The condition is called porogamy. Okay, so you can see the pollen tube growing and then finally entering, okay, the ovule, right, and then into the embryo sac through the micropyle, and the condition is called, okay, porogamy. Now we'll see a little enlarged portion of the embryo sac. Okay, so the embryo sac here, okay, and here you will find the synergid cells, right, okay, and the synergid cells, some finger-like projections, okay, and they are called the filiform apparatus. So it is believed that, okay, they produce some kind of chemicals that actually help in, okay, guiding the pollen tube, okay, towards okay, the embryo sac. So here also you will find, okay, filiform apparatus, okay, right, and you may find, okay, the egg cell here, and there are, okay, the polar nuclei, and here you will find the antipodal cells, antipodals, okay. So this is right towards the chalosyl end, and this side is the micropylar end, okay, and here you can see, right, the pollen tube finally has come up, okay, and then it enters, okay, one of the synergid cell, okay, and I already told you, okay, so this one, one of the synergid cell, and into that one of the synergid cell, okay, the pollen tube enters, because the filiform apparatus, okay, chemotropically they attract the pollen tube. So this is the, okay, pollen tube, and this has the male gametes, okay, and the tube nucleus. So here is the tube nucleus, okay, and here these two are the male gametes, okay. So once it comes into, you know, what I call the synergid cell, right, the pollen tube breaks open, the pollen tube breaks open here, and then it releases the male gametes into the cytoplasm of the synergid cell, okay, and by this time the synergids might have undergone the degeneration, okay, right. So here the synergid, okay, the synergid cell degenerates, okay, the synergid cell degenerates, and here the pollen tube, will release the male gametes, okay, into the cytoplasm of the synergid cell. Okay, the pollen tube, okay, breaks open and releases, okay, male gametes into the cytoplasm of, okay, synergid cell, into the cytoplasm of the synergid cell. Okay, so once the male gametes are released here, okay, the sperms, in fact, okay, what I call them, right? So these are the male gametes here. The male gametes, one of these male gamete will move towards the, okay, egg cell and gets, okay, fused with it. So this is the egg cell, and that results in the process called syngamy. And one more male gamete will move towards the polar nuclei. So here the two polar nuclei, okay, fuse to form a secondary nucleus. Okay, so in this case the secondary nucleus will become diploid. So now, okay, so this all became, okay, a single right, so single nucleus, that is called secondary nucleus. And one more, okay, sperm move towards this, okay, and then, okay, it fuses with this, right, and that okay, results in triple fusion. Okay, the secondary nucleus, okay, plus one male gamete, which will result in, okay, formation of something called primary endosperm nucleus. It's called PEN, right? So this is the central cell, okay, right, and this central cell will be becoming something called, okay, the primary endosperm cell. Okay, so this is what we call the PEC. So that becomes a right primary endosperm cell. Okay, where when the one of the male gamete fuses with the, okay, secondary nucleus. Okay, so this is the secondary nucleus. So two polar nuclei fuse to form a secondary nucleus. Okay, to that one of the male gamete, okay, fuses, and that results in, okay, triple fusion, and that will be the one what we have to call it primary endosperm nucleus. And this whole, okay, central cell will be now called primary endosperm cell. So this is primary endosperm nucleus and this is primary endosperm cell. So once this is finished, okay, the triple fusion and syngamy, okay, is finished, right, the other synergid cell, okay, and antipodals will get degenerated. Okay, so once this is finished, right, antipodal cells and, okay, the synergid cell, they undergo degeneration. So once this is completed, so what remains inside this, okay, primary endosperm cell. So this is what I call the primary endosperm, okay, cell, the PEC. So what remains here, okay, so antipodals, they got degenerated, synergids and all, okay, right. You will find towards the micropylar end there will be the zygote, okay, and here you will find, right, the primary endosperm nucleus. Okay, right. So this is after the fertilization. So what we see inside, okay, the embryosat finally. So now there is a zygote inside it. Okay, the polar nuclei, now it has become primary endosperm nucleus, and this whole, okay, endosperm cell, right, I mean the whole embryo sac cell, it became the primary endosperm cell, right. Now this zygote will, okay, start dividing. So but before that, okay, first the primary endosperm nucleus divides, okay, and after the formation of some endosperm, then the zygote starts dividing. So all that we will see, okay, in the next video as the post fertilization events.